Hello, and welcome to this advanced ChimerMarker webinar on calibrating panels with an allelic ladder. My name is Sarah Copeland, and today I'm going to walk you through the process of using an allelic ladder to calibrate a genotyping panel. This is an advanced topic in ChimerMarker, so if you haven't already watched the introductory webinar, we encourage you to do so before continuing with this webinar. To start, we'll bring in some data files, including an allelic ladder. You may notice that my donor and recipient files are highlighted in blue and green respectively. This is because in the View Preferences menu, under the Chimerism tab, I have input donor and recipient as keywords associated with the file types donor1 and recipient. You can also set the file type manually by simply right-clicking on a file and hovering over Set Sample Type. Now we'll click the green arrow or go to Project Run to bring up the Run wizard. On the first page of the Run wizard, select your genotyping panel, size standard, and standard color, or simply click the corresponding pre-saved template. To create a new template, input a new template name and click the Save button. The second page of the Run wizard includes parameters that govern how size and allele calls are made. The default settings are suitable for most analyses. Be sure that only call alleles in the ChimerMarker panel and Auto-Create ChimerMarker panel are not selected. On the last page, make sure that the Auto-Select Best Ladder and Auto Panel Adjustment are selected. By doing that, ChimerMarker will identify the best ladder for each sample file and use it to create a calibrated genotyping panel that is better aligned to the migration pattern of your data. The data has now been initially processed. The data may look as though it's of poor quality, but this is because many peaks are flagged due to ploidy and heterozygous imbalance, which is expected. At this stage, the alignment of the panel is what is most important. If the ladder sample is in bold, it means that the program was able to align the bins of the genotyping panel to the peaks of the ladder sample, creating a new shifted panel. Navigate to the panel editor by going to Tools, Panel Editor, to save the newly created panel. Observe that there are two project panels. First is the original genotyping panel that was created using information from the kit manufacturer, and the second panel that is now calibrated to the migration pattern for that CE instrument. It can be used in chimerism analysis of any samples that were separated on that instrument under the same parameters. Recalibration of the genotyping panel is typically only needed when there have been maintenance to the CE or a change in the CE run conditions. Save the newly created second panel by clicking on it and then navigating to File, Save as New Panel. Give the new panel a name. After clicking OK, you should see the panel appear in the Panel Templates list. You may also export the panel by right-clicking on it and selecting Export. Now, repeat the analysis using your calibrated genotyping panel for detection of donor, recipient, and shared bins. Newly saved panels are added to the panel drop-down list in alphabetical order. For this run, we want to select Auto-Create Chimerism panel and only call alleles present in Chimerism panel. With these options selected, ChimerMarker will create a project-specific chimertyping panel from the calibrated genotyping panel and will be used to perform single or double donor analysis. You can give a name to this new panel in the field above. Finally, in the last window of the Run wizard, deselect Auto-Select Best Ladder and Auto Panel Adjustment since we've already completed the panel calibration process. Now you can see that the peaks are all in bins, and the only bins that are called belong to the donor or recipient. At this point, you are ready to begin analyzing your data. If you need a refresher on data analysis or reporting options, we encourage you to watch the introductory webinar or one of our other advanced webinars. Thank you for your interest in ChimerMarker. For For more information or for a free 35-day trial of ChimerMarker and other Soft Genetics products, please visit www.softgenetics.com or email info@softgenetics.com. For technical support questions, please write to tech_support@softgenetics.com. Thank you for watching.